we have some assisted reproductive technologies. Okay, so in assisted reproductive technologies also we can go for we can use the genetic engineering. So there things like, let's say, in vitro fertilization. In vitro fertilization means we will go for fertilization in the lab. Okay, so fertilization that happens in the lab, that is nothing but in vitro. In vivo means generally it's in original place. Okay, in vivo and in vitro, these are the two things we'll have. So in vitro fertilization means it's a kind of artificial. So surrogacy, you might have heard about surrogacy. Okay, and then we will have the concept called three-parent baby method. So these are some of the areas where we can further use the genetic engineering. Okay, so here three parents in the sense, like where we will generally use means, see, this is the example for three-parent. So usually certain DNA, we have discussed that certain portion of DNA will get from nucleus, will be present in the nucleus, and certain portion of DNA will be present in the mitochondria. Okay, so mitochondrial DNA would be inherited only from the mothers. Are you getting that? So in some cases what may happen, mothers will have defective mitochondrial DNA. Okay, so in such case what will happen? In such case what will happen means, the child will inherit that defective DNA. To cure that or to treat that condition, what we will do, so first thing is that they will have a father. Okay, so from the father what they will do, they will collect the healthy nuclear DNA. Okay, and then from mother they will collect healthy nuclear DNA. Okay, and there would be third mother with donor mother, from whom they will collect the mitochondrial DNA. Okay, so here from this mother nuclear DNA would be selected, from this father nuclear DNA would be selected, and from this mother mitochondrial DNA would be selected, and they will fuse all these three, and thus they will produce the zygote with the help of this. Okay, so here, as there are three biological parents, the DNA from three parents is involving the baby that has been developed or produced by this technique is known as three-parent baby. Three-parent baby. Okay, so actually in the year 2016 in Mexico, a baby has been produced for the first time by this technique. Okay, so during this process, they had one syndrome called Leidig syndrome. So which was a mitochondrial defective syndrome. So their mother with this syndrome had that mitochondrial defects. So in this case, for the first time, three-parent baby has been developed in the world. Okay, so since 2016, this concept has become popularized. Okay, so that is one thing. So surrogacy is entirely, it is different. So surrogacy means simply will be, yes. So that's where that's where genetic engineering will come into picture. Okay, so they will collect the DNA. Okay, so there in the DNA we will have two types of DNA, like nuclear DNA and mitochondrial DNA. So they will pick the cell, and from that they will take nuclear DNA and they will remove this mitochondrial DNA. So in this process, genetic engineering will come into picture. Okay, so they will segregate this, and here also in this mother also they will have nuclear DNA and mitochondrial DNA, and from that they will pick only mitochondrial DNA. So that means they are cutting that portion only. So there we will get that thing. So then what they will do, so this mitochondrial DNA would be merged with this nuclear DNA of the second mother, original mother, and they'll make mother DNA likewise. Then it would be fused. Likewise. So there genetic engineering will come into picture. Okay. Okay. Is that your doubt? Oh, okay. So that is one thing. So that's how the 3-parent baby can be produced. So that is one another important thing. Okay. So surrogacy means we will have another mother, like sometimes mother might have some issues with respect to uterus. Okay. So there can be some uterine issues. So in such issues, mother might have, like father, healthy father, no issue. So simply sperm would be collected, and from biological mother the egg would be taken. Okay, so then what will happen, this sperm would be fused with the egg. Okay, so they will fuse them, so with that they will produce the zygote. Okay, so zygote would be produced. Okay, so then after production of this zygote, so it would be developed into like embryo. So this zygote would be transferred into the uterus of donor mother. Uterus of donor mother. Are you getting that? So actually here the biological mother may have some issues, or nowadays it became a fashion so as to avoid some postpartum consequences. So now to escape from some sort of postpartum changes, so now it became a fashion. So that's why it became regulated now. Okay. So here you see, like biological mothers, parents would be 2 parents, but thing is that there would be one donor mother whose uterus would be used to develop the baby. Okay, so only that process. In that process, donor mother's soul would be there. Okay, so biological mother, and she would be the donor mother. So that is one. So this is how like surrogacy would be done. So in this process also we have to do, and we have to just go for all these things. So there the articulation can be used. So that is one. So that is another important thing. Yes.
So, in case of repair, maybe instead of going for genetic therapy, so we can just replace the cytoplasm of defective mitochondria.
See, cytoplasm will include all other cell organelles. It also includes the nucleus. Are you getting? So nucleus will have nucleus and their mitochondria. So both will be there. Are you getting? So mitochondria is a part and parcel of the cytoplasm. It would be just floating in the cytoplasm. So we have to specifically replace. We have to correct the defect now. So if we want to correct the defect, then we have to target it. Varna kaise ho jayega? So if you are just replacing cytoplasm, nuclear DNA will get. So then nuclear DNA of the mother, 2 nuclear DNA would be there. Then there is no role of third mother. Are you getting? So then it becomes purely different. So that becomes like IVF kind of method. So where we are taking sperm from one father, the eggs from other mother. So then it entirely becomes different.